Assalamualaikum warahmatullahi taala wabarakatuh, and good afternoon everyone. So today is going to be our first practical for this semester, which is the title of our practical for today is regarding the osmoregulation. If you still remember for our last previous lecture, we are talking about the homeostasis, which is the homeostasis is actually the maintenance of our internal stability in our system, in our body. So for today, we are going to do the practical on the peanut worm. So why do we choose the peanut worm? We choose the peanut worm because the ability of this peanut worm is actually they have the ability to survive in the mangrove, and it is suggesting that it is able to tolerate with the fluctuations to the salinity. So the title for our practical today is the osmoregulation in the peanut worm, Phascolosoma arcuatum, from the phylum of Sipincula. So as you can see and as you can read from your manual practical as I uploaded in your spectrum, so we have the introductions, you can have a look and read about the specimen that we are going to do now. So basically the osmoregulation is the active regulation of the osmotic pressure of an organism body fluid, which the functioning is to maintain the homeostasis of the organism water content. So the osmotic pressure is a measure of the tendency of water to move in one solution to another or by the osmosis. So the higher osmotic pressure of the solutions, the more water tend to move in. Alright. So the osmoregulation also will keep the organism fluids from becoming too dilute or too concentrated. So for today we are going to do on the peanut worm, because the peanut worm is they have the ability to survive in the mangrove, which is suggesting that it is able to tolerate the fluctuations in salinity. So for today's practical, we have two objectives. So you need to do, you need to have two parts of practical, which is the first one. The first objective is regarding, we want to study the ability of the peanut worm to regulate the ionic content in various seawater salinity. And the second part of our practical for today, the objective is to observe the cross sections of the peanut worm. Okay guys, so this is basically the materials that we are going to use for today's practical. So in here we have the rubber dish, we also have the petri dish and the dissecting set. And here is the live specimen that we are going to use for today's practical, which is the peanut worm, as you can see from here. Can you take one or two? So this is actually the, the live peanut worm, okay? And we also have the tissue, we also have the distilled water and 4-level salinity of water. We have the 0, we have the 10, we also have 20 ppt, and the last one we have 40 ppt. And here also we have the aeration pump, we also have the aeration tube and aeration stone. Okay. So to cover the beaker after we put in the specimen, we use the paraphylum, okay, in order to cover the beaker. Then to weight the specimen, we use the top pen balance there. And after we have dissect the samples, we use the dissecting microscope in order for us to observe the integument part in the specimen. Okay guys, so this is basically what we are going to do for our part 1 for this practical, which is the objective for this part 1 is to study the ability of peanut worm to regulate its ionic content in various seawater salinity. So the first method is we need to wash the worm with distilled water and dry the animal gently with the lab tissue. For this practical, we need to use 12 of peanut worm, because each beaker, each level of water salinity, we will use 3 animals. So you need to collect 12 peanut worm in order for us to do this practical one. So after we already wash all the peanut worm, we need to weigh the animals using the top pen balance, which is to obtain the several measurements and to record the results. After we already wash and dry the animal with the lab tissue, we need to weigh the animal using the top pen balance in order to obtain several measurements. So for this practical, you are going to obtain 3 measurements and record the result. Then we need to calculate the average wet weight. Okay, after we already get the average wet weight, we put the 3 animals in each beaker which filled with 500 ml of test seawater. And here we have 0 ppt, 10 ppt, 20 ppt and 40 ppt. And each beaker must have an A stone with gentle aeration and covered with a plastic sheet which is paraffin. And again, we need to calculate the average wet weight of the animals after 12 hours of exposure in the experimental conditions. Okay guys, so we have finished and done with the part 1 of this practical. So we are going to move to the part 2 of today's practical, which is the objective is to observe the cross sections of the peanut worm. So we have the materials here which had been prepared by our assistant science officer. So for the method, we need to dissect the animal using a pointed pair of scissors to retrieve a thin layer of the integument. Okay, and then after you have done with the dissecting of the peanut worm, then we need to observe the integument under the dissecting microscope. Then you need to draw and label the specimen sections. Okay, then after you have draw the table for the result for part 1, so you need to do the discussion and conclusions for part 1. So in this section, you need to write down what is actually you get from the experiment. What is the osmoregulations, what is the process, and based on the in this experiment, what you actually test. So you test for the osmoregulation on the peanut worm, which is to study the ability in regulating the ionic content by using the different level of salinity of the seawater. Or are the marine animals either they are isoosmotic or the hypoosmotic, or are they have the low or high iron concentrations in their body compared to the environment, or how to maintain in the different pressure and etc. So in this section, you need to conclude and discuss on the part 1 result. Then you come to the part 2 result. So for the part 2, you need to write down the method for the part 2. And also you need to draw and label the diagram of the peanut worm, of the dissected peanut worm. Then on the last one, you need to answer the questions that I've given in your practical. Thank you.